the world's first genetically modified humans have been created, it was revealed last night. The disclosure that 30 healthy babies were born after a series of experiments in the United States provoked another furious debate about ethics. So far, 2 of the babies have been tested, and have been found to contain genes from 3 parents. 15 of the children were born in the past 3 years, as a result of one experimental program at the Institute for Reproductive Medicine and Science of St. Barnabas in New Jersey. The babies were born to women who had problems conceiving. Extra genes from a female donor were inserted into their eggs, before they were fertilized in an attempt to enable them to conceive. Genetic fingerprint tests on 2 one-year-old children confirm that they have inherited DNA from 3 adults, 2 women and 1 man. The fact that the children have inherited the extra genes, and incorporated them into their germline means that they will, in turn, be able to pass them on to their own offspring. Altering the human germline in effect tinkering with the very makeup of our species is a technique shunned by the vast majority of the world's scientists. Geneticists fear that one day this method could be used to create new races of humans with extra, desired characteristics such as strength or high intelligence. Writing in the journal Human Reproduction, the researchers, led by fertility pioneer Professor Jacques Cohen, say that this is the first case of human germline genetic modification resulting in normal healthy children. Some experts severely criticize the experiments. Lord Winston, of the Hammersmith Hospital in West London, told the BBC yesterday, regarding the treatment of the infertile, "There is no evidence that this technique is worth doing. I am very surprised that it was even carried out at this stage. It would certainly not be allowed in Britain." John Smeaton, National Director of the Society for the Protection of Unborn Children, said, "One has tremendous sympathy for couples who suffer infertility problems. But this seems to be a further illustration of the fact that the whole process of in vitro fertilization, as a means of conceiving babies, leads to babies being regarded as objects on a production line. It is a further and very worrying step down the wrong road for humanity." Professor Cohen and his colleagues diagnosed that the women were infertile because they had defects in tiny structures in their egg cells, called mitochondria. They took eggs from donors and, using a fine needle, sucked some of the internal material containing healthy mitochondria, and injected it into eggs from the women wanting to conceive. Because mitochondria contain genes, the babies resulting from the treatment have inherited DNA from both women. These genes can now be passed down the germline along the maternal line. A spokesman for the Human Fertilization and Embryology Authority (HFEA), which regulates assisted reproduction technology in Britain, said that it would not license the technique here because it involved altering the germline. Jacques Cohen is regarded as a brilliant but controversial scientist, who has pushed the boundaries of assisted reproduction technologies. He developed a technique which allows infertile men to have their own children, by injecting sperm DNA straight into the egg in the lab. Prior to this, only infertile women were able to conceive using IVF. Last year, Professor Cohen said that his expertise would allow him to clone children, a prospect treated with horror by the mainstream scientific community. "It would be an afternoon's work for one of my students," he said, adding that he had been approached by at least 3 individuals wishing to create a clone child, but had turned down their requests.